Today, our topic is Process of Transcription in Eukaryotes in Molecular Genetics, Unit 2, Chapter 5. Already, we have seen the transcription and the process of transcription in prokaryotes. And today, we're going to see the process of transcription in eukaryotes. So, in eukaryotes, there are three RNA polymerase, but in prokaryote, only one RNA polymerase that is dependent on DNA. But here you have three RNA polymerase in the nucleus. RNA polymerase 1 transcribes the ribosomal RNAs, that is rRNAs, 28S, 18S and 5.8S. S is the Swedberg unit. And the RNA polymerase 3 is responsible for transcription of tRNA. It contains 5S rRNA and snRNA. The RNA polymerase 2 transcribe the precursor for mRNA, precursor of mRNA, and the hnRNA, that is heterogeneous nuclear RNA. So, in eukaryotes, there are three types of RNA polymerase going to take part. So, the one is RNA polymerase 1 is, that is, transcribes. It going to transfer, that is, it going to transfer rRNA, that is 28S, 18S and 5.8S. And the RNA polymerase 3 is responsible for transcription tRNA, 5S, rRNA and snRNA. The RNA polymerase 2 is the precursor for mRNA. The heterogeneous RNA, hnRNA, that means heterogeneous nuclear RNA. So this in this picture you can see the RNA polymerase and a chain is forming. The chain, you can see the cap, that is it acting as a capping, and this is the intron and exon, and the introns are removed by splicing. Okay, splicing, and how the polyadenylation, that is the tail, is going to form. So these are the process you're going to see in the transcription in eukaryotes. So as we have discussed about polycystonic and monocystonic. So polycystonic, the chromosomes are involved and it produces more number of amino acids, that is protein synthesis, which is take place in the prokaryotes. But here in eukaryotes, we are talking about the monocystonic structural gene. So the coding that is the coding sequence. Here the coding sequence is exon. So you can see the coding sequence is exon, and the non-coding sequence. So this is the non-coding sequence. So only the exon is going to form, and this is the non-coding sequence. Okay, that is called intron. So exon is the coding sequence and intron is the non-coding sequence. So the introns are removed by the process called splicing. So you can see the RNA splicing. So this RNA splicing, what it do means, it removes all the introns. Okay, all the introns. And the hnRNA undergoes additional process called capping and tailing. So you can see the capping process. Cap means putting a cap like this. Just think of like that. Capping. And you can see the tailing also. The tailing is adding of tail. Okay, think like that. So cap means putting cap, tail means adding of tail, like that. So we will talk about it. So in capping, an unusual nucleotide, methyl guanosine triphosphate, is added at the 5' end. So this is GATP is added at the 5' end. So GATP is added at the 5' end in capping. Okay, and an adenylate residue of 200 to 300 poly A is added at the 3' end. So here you can see the 3' end, that is polyadenylation. is that is in the 3 dash end. So in 5 dash end, GTP, that is GTP is added, GATP is added, but that is capping. In tailing region, polyadenylated residue is added, that is poly A in 3 dash end. The processed HNRNA is now called as, so this HNRNA after the adding of GATP and poly A, now this HNRNA is called as mRNA. Okay, mRNA, and now it is transported out of the nucleus for translation. So from the nucleus it is that is the that is the heterogeneous nuclear RNA is transported from nucleus to the nucleus, out of the nucleus, transported out of the nucleus. For what it is transporting? For translation process, that we will discuss after completing the transcription. The split gene features of eukaryotic genes almost entirely absent in prokaryote. There is no split genes, but here we are going to talk about the split genes. So the split gene is the special gene which is seen only in eukaryote, not in prokaryote. So each exon have coded to a single polypeptide chain with specific function. So each exon will coded with the polypeptide chain. That means it's going to form the protein, and it has a specific function also. So the exon arrangement and removal of a intron is flexible. So only you can see, so if introns are removed here, only the exons will be there, and it will going to code for the amino acids. And it going to code for the amino acids. For amino acids, what is needed? Polypeptide chain. That is, polypeptide chains are needed where the new genes are carrying. So the single gene can produce different proteins by arranging the exons in different ways. So alternating the splicing patterns, and they have different functions now. So that is. So this is the arrangement of exons. So if it arranged like this, the new proteins will be formed with different functions. So the introns would have arisen before or after the evolution of eukaryotic genes. So when the introns had arise in the protein synthesis, it may be arise before the protein synthesis, that is before the exons are formed, or after. So that wise their function is not clear, because they had removed from the protein synthesis through RNA splicing process. So the introns are called mobile DNA sequence. So they are mobile DNA sequence. They are moving, and they can be spliced, and they can be joined. Okay, so they are acting as a transfer material which is called a transposon, mobile transposon element. So mobile transposon element is acting as an intron. Okay, and they have an horizontal, in intron they have an horizontal gene transfer, HGT. So what is the function of, where it is occurred, and what is the function of HGTP means? It is the lineage of prokaryote gene. That is, the intron is the one of the function, that is from prokaryote to eukaryote it is formed, HGT. HGT, the eukaryotic cells. So between the eukaryotic and prokaryotic, it is a lineage. The HGT occurs in between them. And now it is hypothesized the major role, that is the evolution of the earth have been played, that is earth can be life, how the life was evolved on the earth can be played by HGT, that is HGT, horizontal gene transfer which takes place in the introns. So the introns are formed when the earth is formed when the earth where the life is formed, that time the introns have formed. But now in the eukaryotes, they doesn't play any role. Only the exons are playing for that is for coding the amino acids to produce the polypeptide chain, and from that the proteins are produced. Thank you for watching the video. Give a thumbs up, share and subscribe to Science Easy Tech channel.